Hello everyone, this is Rohan Meshram, and let us continue our discussion on ab initio protein structure prediction. So in our previous lecture, we discussed the steps that are involved in Monte Carlo algorithm that enabled us to generate a lot of decoys. We also learned that comparison of a random number r to the exponent of the delta e term, we can climb up the hill. We can climb up the energy hill using small baby steps. Okay, so this comparison of the energy term, this feature enabled us to cross such energy barriers in our energy landscape. If we don't include such a feature of comparison with r, the random number r, we might end up only moving down in the energy landscape. Okay, and we won't be able to move up. So as discussed, it might appear that that there will be a lot of random moves that are necessary to be taken, and that might eventually make the process computationally quite intensive. Now, in order to minimize this, Simon and Baker use some tricks to minimize the Monte Carlo searches to be performed. Okay, so today we will first discuss what are the tricks that can be used to minimize these Monte Carlo searches, and then we would discuss what are the features of the scoring function that was developed. Okay, so the three tricks that are known to minimize the Monte Carlo searches involves multi-starting, it involves simulated annealing, and using the protein fragments. Okay, so let's discuss how these trick will help us minimize the number of Monte Carlo search. Okay, so to begin with, this the very first approach that is usually implemented in minimizing the Monte Carlo search steps is called as multi-start approach, using this multi-start approach. So here we start out with multiple searches at different places in the configuration search space. Okay, so let me give you an example uh, in order to understand this concept. Say you want to find out the highest elevation on Earth. If you have been given a task of finding out what is the highest elevation on Earth, and you yourself did the Monte Carlo search, you yourself become an agent, and say you are in Pune now, and it is some 560 meters from the sea level. Now you start to wander around, and every now and then you climb up the hill and get back down. Stop, drink some coffee, and start again. Now the question is, could you wander enough and find the highest point on Earth? Maybe you can wander around and end up on top of Mount Karsubai, the highest peak in Maharashtra. But probably you won't get over some mountain ranges in Eastern Ghats in order to even reach near the Mount Everest, which is the highest peak known so far. Okay, so now in order to deal with this situation, what we do now is that instead of having a single hiker who is looking around for the summit, we deploy a bunch of hiker to start out with. Okay, we can now start with whole class and start wandering simultaneously. That is, start from a single point and move each hiker in all direction. Now it is possible that we can find highest peak in Eastern Ghats now. Okay, and this would enable you to do things fast. Okay, so what we can alternatively do is that instead of spreading the hikers right from one place, what we can do is that we can start each hiker at some different place. Maybe drop hikers at different longitudes and latitudes on Earth. Now here is the chance for you to end up on top of Mount Everest. Okay, so this is exactly what we do in multi-start approach in Monte Carlo searches. Okay, so what we do is that we restart from a new random state with a new random path. Okay, so this is what we do. Instead of following a single path to reach the global minimum, what we do is that we begin with multiple starting point, maybe something like this. Okay, we restart from a new random state with a new random path, and hence it becomes more efficient than following a single path. Okay, so this also decrease the chances of algorithm to stuck into well say configuration in the local minima. Okay, so you can kind of avoid that. Okay, so well say for example, if we start out with multiple states, okay, say if one of the configuration got stuck in this local minima, so there would be another state over here or over here or over here, uh, this another configuration that can follow some different path to reach the global minima. Okay, so this eventually brings down the number of Monte Carlo searches that are needed to be performed. Okay, so this is one of the approach. The approach being restart from new random state with new random trajectory. This is one trajectory, this is another trajectory. Okay, so it would become more efficient by doing things simultaneously than following the same path for a given protein. Okay, this would eventually decrease the chances of any protein to get stuck in local minima, and you can achieve something called as the global minima, or the minimal energy state or the native state of that protein. Well, this is one of the trick that can be used to decrease the number of steps that are needed to be performed in Monte Carlo searches. Okay, so this is one of the approach, and let us talk about this next approach called as simulated annealing. Okay, we need to play around with adjusting the temperature in this simulated annealing approach. Okay, now to understand this, let us go back to let us go back to our MC Monte Carlo algorithm, where we proposed this term, that is e raised to minus delta E by kt. So this e raised to minus delta E by kt term is eventually the factor in Monte Carlo algorithm that decides if or not to keep the high energy confirmation. Okay, so now let us see what will happen if we keep the high temperature. Okay, now uh, since temperature is present in the denominator position in this particular term, therefore if we keep the temperature at higher values, or if we keep high value in the denominator, the overall value of this entire term, the overall value of this entire term would be small, right? Now again we are taking exponent of this small term. Okay, so since we are taking the exponent of this small value, and if we follow the basic calculus, exponent of a small number will always be close to 1. Now we know that if the value of this term approaches to 1, we need to accept the configuration. Therefore, at high temperature, we shall accept, we shall accept every move. That is, we can move around in the energy landscape with minimal energy barriers. Okay, so that is what I wanted to say. Okay, so eventually this is what is happening. In short, and conceptually what we are doing is that by playing around with this temperature, we are trying to squish down the energy barrier so that we can move around easily in the energy landscape. Okay, so this is what we are doing. We are trying to squishing down this energy landscape so that we can easily move around in our energy landscape as such. Okay, therefore if we maintain high temperature, it will allow us to take larger steps. Okay, so maintaining high temperature will force the algorithm to accept every random move that we make, and this will allow to move fast in the conformational space and thereby reduce the Monte Carlo steps. Okay, so this is the second approach that can be used that can be used to reduce the Monte Carlo steps that are involved in our algorithm. Okay, so now this is what would happen when you increase the temperature, right? So increasing the temperature would enable you to move fast in the conformational space. Okay, now let us think of another scenario when we decrease the temperature instead of increasing it. Okay, so as the temperature drops down, the value of this term e raised to minus delta E by kt shall be close to 0. Now, if it is close to 0, that would means that there is tendency to reject high energy conformation. That would mean we would accept only low energy state and hence move down in the energy landscape. So, low temperature shall force trajectory towards low energy basins. Therefore, proper tuning is required in temperature for fast Monte Carlo calculation. Okay, so in order to achieve this, all we need to do is to set an adjust temperature feature during bookkeeping stage of the algorithm. Okay, so this is how you can minimize the steps that are involved in Monte Carlo searches. Now the third approach to minimize the Monte Carlo search is to use known protein fragment. Okay, so now this is based on the argument that sequence allow a relatively small number of local structures. So what if we found out the local structure from PDB whose sequence is similar to random fragment and use those in our Monte Carlo searches? Okay, now isn't it cheating? So by definition, ab initio means we are expected to start from scratch, and here we are proposing to use known fragments from already known structure. Then how is it ab initio prediction at the very first place? Okay, and people that use such a kind of using known protein fragment approach, they formulate a new term and they say that this is called as de novo modeling approach instead of ab initio. Okay, so whatever it is, this is what we have summarized so far. Okay, so we have learned how Monte Carlo algorithm works, and we have figured out some smart way by using which you can make a good structural candidate, or you can generate a lot of structural decoys. Okay, so so far so good. We figured out how to get good folded structure. Okay, now there is other important aspect of ab initio concept that is still left in our discussion. Okay, we have only dealt with how do we generate lot of structures. Okay, but we also need to figure out how to score the folded structure, or how to find out which one is the better folded structure as such. Okay, so here is well to begin with this concept, I think you should first know what is centroid representation, right? So well, let us begin our discussion. Most of the protein folding concepts that we were dealing till now, or whatever we were discussing till now, well, these concepts focused mainly on the backbone in your protein. You know, the fold of the protein is usually associated with the backbone. Okay, so for time being, or for time that we were discussing so far, the major objective was being recognized how protein would fold, rather than focusing on rather than focusing on the atomistic details of the model. Okay, but while scoring whatever fold that we have generated, well, side chains must also be considered. Okay, you cannot completely rely on the backbone while it comes to the scoring the entire model. So the side chains are needed to be considered here as well. Okay, so the problem is that we do not want to ignore the side chain. At the same time, we wish to avoid the heavy atomistic calculation as well. Okay. Now Simon and Baker did a trick to get out of this tight corner. They made a new representation of the protein that don't contain all atom and represent the side chain in a simpler way. They call such a simplified representation of protein as the centroid representation. Well, now let us talk about this centroid representation first. Now I told you ki why do we need this centroid representation? Okay, so now in general term, you can call it as an approach for coarse graining. Okay, in molecular modeling area, what we do is we combine a bunch of atoms together and represent them in a unified fashion. We say that we have performed coarse graining. Okay, so it is necessary to have atoms from the backbone, because if we want to fold the protein, we will have to manage the phi psi angles when we take the random moves in Monte Carlo algorithm. Okay, so we need something that would briefly that would briefly summarize the side chain, as well as let us have the detailed conformational space that is being generated by twisting and tweaking those phi and psi angles. Okay, so what these guys did was to draw a sphere of radius that covers the entire side chain, as you can see over here. So now this sphere will be considered as a single entity, the side chain, or simply we would call it as the R group. Okay, so now this R or side chain will have a specific volume and specific position around the protein's backbone, based on what amino acid residue that are represent. For example, the volume of this R or side chain group will differ when you consider valine and alanine as your amino acids in your protein. Okay, so in this way, we have all backbone atoms and side chain atoms in centroid representation. So this fix consider side chain in a protein representation and decrease the computational cost as well. Okay, so what we are doing over here is that we are trying to figure out certain representation that would include the side chain, but also enable us to have flexibility in the backbone as well. Okay, so this is called as coarse graining. Okay, now well, this is what we have already discussed. Okay, uh, so let us talk about this centroid representation now. So after considering the side chain as a single entity, what they did next is that they tabulated the position of your R given the location of backbone atoms, that is N, C alpha, carbonyl carbon and carbonyl oxygen. That is, they tabulated parameters for calculating the bumps. Okay, for example, distance between this R and N. What is the distance between this R and this C alpha carbon atom? What is the distance between this R and carbonyl carbon atom? What is the distance between this R and carbonyl oxygen atom? All of this has been done, or you can say all of this, in order to calculate an average distance. This R can get close to any atom from the backbone. Okay, the logic being to know how far this R is to be kept away from the backbone in order to avoid the steric clashes. Okay, so this representation was used in Simon and Baker approach, and this approach allows to move fast during the calculations that are involved in Monte Carlo searches. Okay, so this is how they ended up creating a simplified representation of proteins for their algorithm. But it would complicate the matter if the physics based energy functions are to be used for energy calculation. Okay, so these physics based energy function need to know what is the van der Waals contribution, solvation energy, etc. for each component in the model to compute the free energy. And we have represented the side chain in centroid representation in our models, right? Therefore, it will be very difficult to calculate or state these components like van der Waals contribution and solvation energy. Okay, so in short, it is not possible to use general physics based energy function directly as they are over here. Okay, they so what they have done is that they have a modified version of this concept where they have parameterized the value statistically. Let us not go in details of parameterization process here, as these concepts are far beyond the scope of discussion here. Okay, so if you want to go in more details, you can refer the second paper that is published by Simon and Baker in 1999. Okay, so in this paper, Simon and Baker describe as to how these structures can be scored. Okay, and that is why these methods use the term score rather than energy while describing good folds and bad folds in their papers. So, in short, using this centroid representation, well, it becomes handy during calculation. Well, say for example, if we decide to use valine in our calculation, well, say for example, let me draw this valine for you. So we'll say for example, let us draw this backbone. We have this nitrogen, we have this carbonyl carbon, carbonyl oxygen, then we have this C alpha carbon atom, right? And if we are trying to use valine, then we would have C beta carbon atom. Okay, then you have got C gamma 1 and you have got C gamma 2 carbon atom. Okay, so if we try to number, or if we try to count out how many atoms would be there if we try to include this valine, we would be end up with 1, 2, 3, 4, 5, 6 and 7. We would have, in short, using centroid, well, so for example, if we decide to use valine in calculation without centroid representation, we will have to deal with 7 atoms. And if we use this centroid representation and considered your entire side chain as single entity R, then we would have to deal with only 1, 2, 3, 4 and 5 entities now. We have to deal with only 5 rather than dealing with 7. So this would enable you to have faster calculations. Okay. So this is what your centroid representation is, and this is why it is essential that we need some abstract way to represent the side chains, and hence we ended up using this centroid representation. Okay, so the idea was to prepare a scoring function that will discriminate good folds from the ones that are not very good. Okay, so their approach was to use information of structure from the PDB and derive a scoring function. Now you should remember here that here we are using the structural information to score the fold and not to create the fold. Okay, so there are a whole lot of structures in PDB. And now the idea is, why don't we use this wealth of knowledge, use this knowledge of what we know about proteins and come up with something to score and at least imitate what nature does. The idea is to use database of structure and come up with knowledge that will use to recognize a good fold from a bad fold. Okay, so now this is termed as process of generating knowledge for your scoring. Okay, so this process of generating knowledge for scoring entails three steps, or you can say that the process of generating knowledge for scoring is a three-step process. Okay, so the first step involves something called as the feature identification. So what we are doing is that we are generating a bunch of configuration and look at them and ask question like, which of these structures really look like a protein. That is, we need to identify certain features that may contribute in distinguishing proper fold from a non-native fold. Okay, for example, if we end up generating a configuration that is whole lot of spread out, then it's not probably a good fold, because from existing structure we know that protein folds are compact. They tend to be compact. Okay, now we can consider hydrogen bonding as another feature. Okay, we can check the distribution of hydrogen bonding pattern all over the proteins or in specific secondary structures, right? Alternatively, we can check if polar and non-polar residues are properly distributed all over your surface in your structure. Okay, from the existing structure we know that non-polar residues prefer to present at the core, while polar residues are usually found on the surface. In this example, now we have selected two features in this way for scoring. That is, we have selected hydrogen bonding as a feature for scoring, and we have selected residue distribution for scoring the fold. Okay, so the next thing, or the next step in generating this knowledge for scoring, is that we choose appropriate variables to describe this feature. Okay, we might generate some accessibility profile for determining the residue distribution. We might create hydrogen bonding profile in a similar way. The overall idea is to quantify those features in order to use it for scoring. Okay, and thirdly, we will definitely have to evaluate the effectiveness of whatever features that we have quantified alone and in combination. Well, say for example, we would like to evaluate how well hydrogen bonding and residue distribution works alone as well as in as well as if they are used in combination with each other. Okay, so now all of these features like hydrogen bonding and amino acid residue distribution, all of these features contribute the sequence dependent terms. Why? Because these features are controlled by what is the sequence of amino acid residues that is present in your protein. Okay, so now, therefore, we term such features that are dependent on sequence of amino acid residues as sequence dependent feature, and we have discussed two of them. So, along with sequence dependent term, Simon and Baker also used some sequence independent terms like secondary structure packing, bumps, and compactness. Okay, so here they look in protein data bank how often these secondary structures are arranged in experimental protein and correlate with fold you get and score your folds accordingly. Okay, say for example, if there are 6 strands in the structure, okay, then check out in PDB on how often these 6 strands are arranged. Okay, well, say for example, if you end up with say these 6 strands, right, and what you do now is that you try to figure out how these 6 strands are usually arranged. Well, say for example, do these 6 strands form two beta sheets, one beta sheet containing 4 strands and another beta sheet containing 2 strands, or do it happen the other way around? These 6 strands are distributed in two beta sheets in the form of 3 strand in one sheet and 3 strand in another sheet. Okay, so this is how you can correlate. Okay, so it seldom happens that the protein acquires a strand as a secondary structure and there is no other strand near it to partner the hydrogen bonds. We know that if the structure forms a strand, then it must be stabilized by neighboring strands, as we know that these beta sheet have intersheet hydrogen bonding, right? So in short, if your algorithm ends up identifying a single strand, okay, if your algorithm ends up identifying only one strand, that means that something is not right and the identified fold thus should be scored low. Well, now, if there are helices in your configuration, then check in PDB as to how they cross. Okay, that is, we are supposed to consider helix relative geometries. Okay, so it is known that there are certain patterns of helix orientation that are more preferred over some other. Okay, so all you need to do is to correlate these secondary structure packing elements. Okay, like helix relative geometries and strand rearrangement in order to score your fold, whatever you have created as a good fold or as a bad fold. Okay, uh, next feature can be in form of bumps. Okay, now you know that two atoms in your protein cannot occupy the same space. Okay, no two atoms in your protein can come close enough than the sum of their van der Waals radii. Okay, that is the two atoms do not occupy the same space, they do not overlap. So, based on this van der Waals distance of atoms and of the and that of our centroid, right? If you end up generating a configuration that has overlapping atomic coordinates, you need to score it low or even reject such configuration, right? So, it is another feature that can be used to distinguish a good fold from a bad fold. Okay. Now, compactness can be described by radius of gyration. Okay, so uh, as I have already described, that compactness is also one of the factor. We know by studying a lot of proteins that have been crystallized or experimentally derived so far, we know that this protein tends to be compact. Okay, and this compactness can be attributed mathematically in terms of radius of gyration. Okay, so these lower values of radiation of this radius of gyration means that the fold is compact. Okay, now what you need to do is that you need to quantify how often that happens in PTB. Okay, and correlate with how often that happens in your structure that comes out of your Monte Carlo algorithm. Okay, you would need to quantify that and then decide if the configuration is fit or if it is not fit. Okay, now do you remember that do you remember that this term again, e raised to minus delta e by kt? Okay, so this function usually determines if or not to accept or reject the new configuration in the Monte Carlo algorithm. Okay, so in this expression, the value of delta E is based on this sequence dependent and sequence independent term that we calculate in scoring. So in this way, my friends, scoring function usually guides the search process. Lower or higher energy scores obtained while scoring will dictate the algorithm how to proceed in Monte Carlo steps. So it is so this is how these two components work together hand in hand. These two components, like scoring and creating new poses, do not work independently of each other. Well, it don't happen like the algorithm first create all the decoys and then it would score whatever decoys are produced. It don't work that way. Okay, so so instead, our algorithm, these two components work alongside to make things happen. Okay, so this is how your scoring and post generation are intertwined with each other, and they work simultaneously. Okay, your scoring is dependent on what kind of fold you have got, and your score would eventually determine if or not to use that fold or not for a next Monte Carlo search, or to begin or to anneal with that next Monte Carlo search. Okay, so this is all I have for you guys, and I hope you enjoyed this lecture on ab initio structure prediction and eventually the entire structure prediction as a field as such. Okay, thank you for your kind attention.